Okay, so today is our last lecture, and we are going to just go through a short lecture to talk about biotechnology and specifically about genetic modifications. So what do you think of when you think of genetic modifications? Because when we say that, lots of people have their own ideas as to what that means. They bring their own experiences to the table on that. So for some of you, you may think about GMO food sources. That's a hot topic a lot of times. Others may think gene therapy, cloning of organisms, maybe even cloning of people. So some of you might go down the science fiction route and think of movies where we have human clones all being, controlled by some individual. So lots of people have different things that they come to the table with when we talk about genetic modifications. So we're going to talk about what it really means, and then you can think about the implications thereof. So let's just start with some definitions so that we're all talking the same language. So first of all, biotechnology, what is that? It's basically just the use of a biological system to create a desirable product or end goal. Genetic engineering is modifying the genome, or a genome being like the whole set of DNA of an organism. So genetic engineering is modifying that genome to improve its characteristic or to make a particular product. So the next definition is genetically modified organism. So that's that GMOs that we talked about. And that's an organism whose genome has been modified in some way. So I will go ahead and give you a spoiler alert. GMOs have been happening way longer than you think. It seems to be the hot topic right now, but even Native Americans that lived in the US and also throughout the world really, when we talked about the corn production, corn by itself does not produce these large cob that you use to eat. Those seeds originally are very small, but when you crossbreed plants and you take the ones that have the largest kernels and you crossbreed them with the most hardy corn, then you get a genetically modified organism because we are selecting for a particular set of genes. Okay, so genetically modified is not unique to the 2000s. Okay, and then there's transgenic organisms. That is a particular type of GMO that has a gene from another species inserted into its genome. Again, this can be manipulated by people in order to make new types of food in particular. There are also all kinds of other things like our gene therapies. But this also happens naturally. Sometimes when you have insects that are eating from different animals, they can insert DNA from one organism into another. So even transgenic organisms occur without the help of human input. Okay, but now we're all on the same page with definitions, so let's move on. So let's start by talking about cloning. So what do we even mean when we say that? So cloning is the term that describes several different processes that are used that produce a genetically identical copy of a biological entity. And the reason I say biological entity is because you can clone an organism, you can clone DNA, or you can clone a cell. And those are the types of cloning that are listed below. But whatever the copied material is, that is referred to as the clone of the original. And so we're going to just take a quick look at some of those at the moment, just so that you know what they are, how they're accomplished. So let's get to it. Okay, organismal cloning. So organismal cloning is going to involve asexual reproduction. So we know by now that that involves mitosis, does not involve meiosis. And so this has actually been going on for forever practically in the horticultural industry. In fact, you may have done this yourself. I have. for instance, with hydrangeas, I can produce a whole new hydrangea plant by just taking a portion of the mature hydrangea plant that I have in my yard. And the way I do that is I take a clipping and I can take off the leaves and I bury the part of the stem right here into a pot, and it will begin to grow roots and create an entirely new organism. So the way that that is accomplished, because as you know, this shoot right here doesn't have roots already. Well, each of those cells, just like we've talked about before, all of the cells in your body have the complete complement of DNA for every part of your body. Same is true in plants. And the genes that are expressed, like we discussed recently, are either turned on or turned off based on hormones that are produced. And so when I am cloning my hydrangea, for instance, I can just put it in loosely packed soil, and the plant knows immediately that it's going to need to grow roots or it's going to die. And oftentimes when we are cloning, we take lots of different clippings and only some of them survive. The way to make sure that we get all of those clippings to work is that we can dip that clipping into what we will call root tone, which is a powder that contains hormones that induce the plant to grow roots. Okay, and so by doing that, we are cloning the original organism. So this has been used in horticulture for a very long time. Okay, so organismal cloning in animal cells is a little bit different, and we call this reproductive cloning. So this is where we start to get into ethics, and people get very nervous about cloning animal cells because that means you can also clone human cells. So today I'm not going to talk about the ethics of it. I'm going to let you think about that and do your own research. I have definite opinions on it, but I want you to do your own research and see what you think. I am just going to tell you the method at the moment for organismal cloning in animal cells. What we do for that is we would take a donor nucleus from a somatic cell, so not the sex cell, so it would be diploid, and then we also take a donor egg cell. And what we do with the egg cell is we remove the nucleus. So it's just a cell with no nucleus. And then these two cells are fused together. So now we have an egg cell that has the nucleus from our donor cell. And then those fused cells begin to divide as they usually would, producing an embryo. And that embryo is implanted into a surrogate mother, basically. And it's using the natural hormones of the mother to develop that embryo into a full organism. So that is how we clone an animal. So the second type of cloning is molecular cloning. And this is used to assemble what we call recombinant DNA. And we'll talk about that in a minute. And then we're going to ask a host organism to make multiple copies for us, right? Multiple clones. And this method takes advantage of the fact that DNA is fundamentally the same in all living organisms. Now we've talked about the variations with across species, but fundamentally it's the same structure. So there are 7 steps to it, and I'm going to walk you through those, and then we're going to look at a diagram that shows how these are done. So the first is that we have to choose a host organism and a cloning vector. And what they mean by a vector is basically something to carry the DNA that we're interested in. So then we're going to prepare that vector DNA, and then the preparation of the DNA to be cloned. We're going to create a recombinant DNA, which means putting those two DNAs together, the host DNA and the vector DNA. And then we're going to introduce that recombinant DNA into a host organism. And that host organism, we're going to use the DNA replication machinery to allow it to replicate that DNA over and over again. And then we're going to select for the organism containing that recombinant DNA, screening for clones with that DNA insert. Okay, so here in our example, let's assume that there is a piece of human DNA that contains the gene that we want. In this case, we are saying we want the gene that makes insulin. Okay, and so what we're going to do is we are going to find a host, which in this case is our bacteria, and the reason we would use bacteria is because it multiplies very quickly. If we waited for other cells, we might have to wait a month to get the protein that we want, but bacteria divides every couple hours sometimes, so we can produce a lot of what we need. Bacteria also contain what we call plasmid rings, which is that excess DNA, and it gets replicated as well. So what we're going to do is we're going to come in and use restrictive enzymes, which basically cuts this vector out into a straight line. And then we're going to use DNA ligase, which you have talked about before, which allows for this insulin gene to be inserted into the plasmid, and that ligase is going to bind that DNA together. So now we have what we call recombinant DNA. Recombinant DNA means that it has a portion of the host DNA and the desired DNA fragment that we are looking to reproduce, recombinant DNA. And then our host cell takes it up, so our bacterium, and we allow that to grow over time. And then that bacteria, as it continues to replicate, is going to also replicate the recombinant DNA. And so we have gene cloning that occurs. So these bacteria continue to replicate the desired gene. And in our case, that bacteria also produces the product that we want, which is insulin, because it has gone through gene expression as well. So that is molecular cloning. Our third type of cloning is what we call cellular cloning, and this is exactly what it says. It's the cloning of a cell that with the means to derive a population of cells from that particular one. It's very similar to embryonic cloning that we talked about before, or reproductive cloning. However, its intention is to develop just a population of cells that we can then use for other purposes. So the method is called somatic cell nuclear transfer, which is what we did with the organismal cloning as well. Okay. So here we go. These are a few things to look at. So our normal development here on the left, we have sperm and egg come together for a zygote. They form what we call a blastocyst, which is that first cell, which then divides and becomes an adult. Our reproductive cloning that we talked about before, we have our adult egg, our denucleated oocyte that come together, and the rest of it proceeds from there. Our therapeutic cloning or cell cloning is the same as reproductive cloning up to about the blastocyte or blastocyst. Then we begin to produce embryonic stem cells, and essentially stem cells are cells that will continue to divide, but they aren't differentiated until they come into contact with certain hormones. So we can take these undifferentiated cells and essentially make different tissue types that we might need. Red blood cells, skin cells, and this is used in things like, for instance, if we have a burn victim, we may use these stem cells in order to produce new epithelial tissue for them. So that's where cellular cloning comes in. So here are just a few things to consider when we are discussing the ethics of cloning, and these are not my ideas. These are, I'm giving credit to Dr. Palmer here. So it looks at first glance that the advantages outweigh the disadvantages just based on pure numbers. However, looking at the disadvantages, you might want to consider the impact of each of those. So this was just to get your brain started and thinking about the ethics of it. So gene therapy is one of those cool methods of biotechnology that utilizes viruses. So viruses will inject their DNA into a host cell specifically for the purpose of being replicated. And so what biologists and geneticists have done is utilize that and inserted a gene that is desired into the viral DNA to have recombinant DNA, and then allow that virus to inject its host cell. Now, there are usually modifications to the viral DNA other than the desired gene, such that the virus is a safe virus. But it's really cool that you can use that organism's ability to then produce good things for people. So our first successful gene therapy was in the 1990s, actually 1990, with a little girl named Ashai Da Silva, and she had a disease called SCID, or Severe Combined Immunodeficiency. And children with that disease lack the enzyme adenosine deaminase. And adenosine deaminase is an enzyme that helps to mature your immune system. So these children that have this disease are susceptible to all kinds of illnesses, and they tend to die pretty early. So for Da Silva, they used a virus to insert a gene that would create adenosine deaminase, or ADA. And so the way they did this is what we call ex vivo. Vivo just means living, so ex being outside of the living organism. And the way they did that was they took bone marrow stem cells, so you know stem cells grow really fast and can be differentiated later. And that stem cell, of course, has the defective gene. And they used a retrovirus, and a retrovirus just acts back. It replicates backwards. So it uses something called reverse transcriptase, and it's genetically replicates with RNA. Okay, so that's just a little background. You don't need to know the details on that. But they use this retrovirus to bring in the normal gene into this stem cell. So it has viral recombinant DNA, and that, RNA. And that viral recombinant DNA carries normal genes into the genome, where it is replicated over and over again. Then we take these cells that are now genetically engineered and reinsert them back into the bone cells, into the bone marrow, where they replicate. And when they replicate, they're also replicating with the gene that's restored for ADA. And so now we have DNA replication happening in stem cells. So we're going to have cloning of that segment of DNA over and over again. So that restores the health to the children. So that is an example of gene therapy. So I mentioned that with Da Silva, they used ex vivo gene therapy, and that's where they take the cells out of the body, they transform it with recombinant DNA, and then place it back into the patient. The other way of gene therapy is what we call in vivo gene therapy. And that means that the gene is cloned into the genomic virus, into the genome of the virus. And when we do that, then we can put that virus directly into the body. We put those cells into the body, they're replicated, and when they are replicated, again, they produce the correct gene. Okay, let's talk about GMOs, genetically modified organisms. So with GMOs, I mentioned that we have been genetically altering agricultural products specifically for a very long time. Well, oftentimes when people say GMOs, they don't just mean genetically modified. Oftentimes what they mean is genetically modified by techniques other than cross-pollination. So in other words, extracting DNA or inserting DNA using biotechnology instead of cross-pollination. So just wanted to say that, but it's pretty ambiguous. So when people say GMOs, they may mean one or both. Okay, so GMOs are any genetically modified organism. So we often times will think of it in terms of our agricultural products, and that's fair because that's where a lot of genetic modification happens. And so, for instance, we do use genetically modified corn, for instance, and we basically modify a bacteria cell and then allow it to infect the whole plant. And what that does is well, one of the most common things is that we've selected for a gene that produces a pest-killing protein. And so when that is inserted into the genome of our corn plants, then those corn plants are pest resistant. And if you know any farmers, or if you've ever tried farming or just doing a small vegetable garden, you know one of the biggest issues with trying to grow a food supply is the pests. Any kind of bug or blight that will come and take away your product. So not only does that mean loss of money for the farmer, it also means a decrease in the food supply. So our GMOs that allow for pest-resistant corn, it actually increases our food supply. Another example of genetically modified organisms is what we have mentioned before, and that is our human insulin gene being injected into the plasmid of a bacterium DNA, and that bacteria then producing insulin, which can then be separated and purified and used for diabetics. So the top 4 agricultural products that are genetically modified are corn, soybean, cotton, and canola. And about 90% of those that are produced are genetically modified. So I'm just going to talk about this real quickly. That is near and dear to my heart is cotton. My family is from South Georgia and Alabama, so a fair amount of cotton, at least grown in the area, if not directly by my family. And so cotton is particularly attacked by pests, like boll weevils, for instance. And it has been genetically modified to resist pests. And you use cotton products every day of your life. Anything from Q-tips to your towels to the clothes you wear all contain cotton. So keeping an ample supply of cotton is incredibly important. Now, having said all that, I am aware that when you are genetically modifying an organism, particularly a food supply, you may have unintended consequences. And I think there have been some of those. I think they've also been overly stated, perhaps. But it is something to keep in mind. Now, one particular genetic modification that when I was in graduate school we were challenged to try to come up with was trying to make coffee that is a decaffeinated plant. So when you drink decaffeinated coffee, it is decaffeinated by a chemical process. And when we looked at all the metabolic pathways that involve caffeine, there is pretty much no way of taking caffeine out of a plant. It cannot grow without that particular biomolecule. So it's just interesting. Some things you cannot genetically modify because it will not give you viable products. Another example of a GMO is when we add what we call green fluorescent protein, or GFP, to an organism. And there's actually a small little inset in your textbook about that. GFP, when you put it behind the promoter of a particular gene, allows you to see where that particular gene is being expressed. So for instance, on this image here, we are looking at roots that have root hairs growing out of them. So that is, this right here is a root hair, so it comes off the side of a root. And what we have labeled with green fluorescent protein here is actin filaments, so part of the cytoskeleton. And this can be done while the plant is still living. So as it grows these root hairs, we can watch how the cytoskeleton, particularly the actin, changes over time. And so it's a very useful tool for biologists to study organisms as they go throughout change. So genetic modifications can be used for that as well. So the benefits of biotechnology are vast. And so we have health benefits from biotechnology, and as the biotechnology boom has increased, we've had reduced rates of infectious diseases, which of course is saving millions of lives, children and adults for that matter. And it changes the odds of serious life-threatening conditions because we can use gene therapy to change it. We can tailor treatments to individuals so that we can minimize health risks or side effects, and we can create more precise tools for disease detection. So those are just the health benefits of biotechnology. Biotechnology also fuels the world. So what you don't realize is all of the biology that goes into things like fuel and manufactured productions and making cleaner fuels and even detergents for your clothing, and also keeping those things safe, keeping your environment safe. In fact, when we've had oil spills in the ocean, the main way that that is cleaned up is through bacteria that actually eat the fuel. So biotechnology is fueling the world and keeping it environmentally safe. Obviously, biotechnology is feeding the world, like we talked about with some of the GMOs, generating higher yield crops, and even using those higher yield crops in a smaller amount of space means we get to use less agricultural space and therefore is environmentally friendlier. And so we're developing crops that have better nutrition, and that's keeping children throughout the world healthy. And I think sometimes we tend to forget that very quickly in the US because we have ample food supply, ample nutrition, but that is not true throughout the world. So what's next? I hope that you have learned plenty in this class to start laying the groundwork for what's to come next. And I hope that you are a part of the solution using biotechnology armed with all of the information that you've learned this semester to move on to the next level, learn more things, create new products, create new ways of doing things, and discovering more things. It's been great to have you along for 111. Hopefully, I'll see you in 112.